Hello and welcome to all my students. So today I am going to start with the next lecture of molecular basis of inheritance, where we are going to study about the mechanism of replication. So far you have seen that what is the structure of DNA, packaging of DNA and search for genetic material, in which you have seen the experiment by Griffith, Hershey and Chase. Today we are going to see the first step of central dogma, which is replication. Beta, replication means copying of DNA to produce new DNA strand. Now, how this thing takes place, how replication takes place, what is the mechanism behind replication, that we are going to study today. But before we start with the mechanism of replication, there was one experiment performed by Meselson and Stahl that explains that DNA replicates semi-conservatively while they were working with E. coli as an experimental organism. So first we are going to check out the experiment by Meselson and Stahl. The aim of the experiment is to show that DNA replicates semi-conservatively. The DNA replicates semi-conservatively. Now, what does this semi-conservatively means? So, before I tell you one more point, let me write while working on E. coli. So, they took E. coli as an experimental organism while working on E. coli, right? Now, before I start explaining you the whole process, let me tell you what is semi-conservatively means. You know, beta, that DNA is double-stranded helically coiled. Be it eukaryotic or prokaryotic DNA, DNA is not single-stranded like RNA, but rather it is double-stranded helically coiled. You have seen the model proposed by Watson and Crick in last lectures. Right? Now, beta, when DNA is double-stranded, both the strands of the DNA, they are anti-parallel. Correct? Right? Now, since both the strands are anti-parallel, both the strands of the DNA will undergo replication. Both the strands of the DNA will become parent DNA, and on both the strand, new daughter strand will be produced. Right? So now suppose these are the two parental strands. Both the parental strand will unwind, and on them new daughter strand will be synthesized in anti-parallel manner following the rules of complementary base pairing. Right? So now this is the parent DNA. From the parent DNA, after replication, you obtained two daughter DNA, and in each daughter DNA, 50% is from the parent and 50% is newly synthesized. That means DNA is replicating semi-conservatively. Semi-conservatively simply means that the new daughter strand produced from parent strand, new daughter strand produced from parent strand has 50% new strand and 50% the strand from the parent. So what are you going to write? Let's see. Let me write it over here again. Semi-conservative replication means that a newly synthesized DNA will have one strand from the parent, right? And another strand will be newly synthesized. will be newly synthesized. Clear? This is semi-conservative replication means. Now, how it was proved by Meselson and Stahl? Let's try to understand the experiment performed by these two people. Beta, first of all, they used the medium for culturing E. coli. They prepared two medium for the culture of E. coli. One which is rich in ammonium chloride in which the nitrogen is doped with heavy isotope, that is N15, and another medium was again ammonium chloride, but this time nitrogen was doped with N14, which is the light isotope, non-radioactive isotope of nitrogen. So basically these two people, Meselson and Stahl, they prepared two medium for the culture of E. coli. To study the method of replication in E. coli, they prepared two medium for the culture of E. coli, right? Now, those two mediums are one ammonium chloride in which nitrogen was replaced by N15. N15 is the heavy non-radioactive isotope of nitrogen. One more medium having ammonium chloride was prepared, but this time nitrogen was replaced by N14, which is the light isotope of nitrogen. Correct? Now, first of all, beta, these two people, Meselson and Stahl, they made the E. coli to grow in N15 medium for several generations. Several generation means, see, in generally E. coli, DNA replicates in 20 minutes and they divide almost like within 38 minutes. So E. coli DNA replication takes place only within 20 minutes. So you can say that E. coli, since this bacteria divides very fast, it is very quick to obtain their new generation. That is why Meselson and Stahl, they preferred E. coli as they produce new DNA very quickly within 20 minutes. So what these two people did, they took a medium having N15 in which they cultured E. coli for several generations. That means these two people, they allowed E. coli to replicate its DNA in the medium having N15 for many generations. What will happen after letting E. coli replicate in N15 medium for several generations? Check this. What will happen? Just a second, let me add a page first. Okay. Now, in first step, this is step number 1 of the experiment. In first step, E. coli was made to replicate in medium N15 for several generations. So now what will happen? Suppose this is my initial parent DNA of E. coli. Right? Now this is made to replicate for several generations in medium having N15. So every time new DNA strand will be synthesized on the parental strand. Now new DNA means new nucleotide. New nucleotide means automatically nitrogen will be required. And that nitrogen to synthesize new nucleotide will be taken up from the medium. The medium nitrogen is N15, which is heavy. So automatically what will happen, beta? For first generation, like for first generation, both the parental strand will undergo replication. Correct? Now, on each strand, the new DNA strand will be synthesized in which nitrogen will be N15 type. Correct? Now, this is first. For several generations, Meselson and Stahl let the E. coli grow in N15 medium. So, after several generations, after several generations, the whole strand of DNA will become N15. Such DNA in which every strand has become N15 is known as heavy DNA. So first step of Meselson-Stahl experiment is to make the E. coli DNA, normal DNA, turn into heavy DNA. Heavy DNA means when both the strands are having nitrogen, which is N15 in nature. And it will happen only when the normal DNA is made to replicate several times in ammonium chloride medium having N15. Correct? Yes. Now, in step number 2, now these people, they took the E. coli rich in heavy DNA and now they changed the medium of E. coli from heavy to light. Now that means they were made to replicate in a medium which is having light isotope of nitrogen. Now, what will happen over here? Tell me. Within 20 minutes, the first generation will obtain. After 20 minutes, first generation of E. coli beta will be obtained. What will happen? Semi-conservative replication of DNA. If semi-conservative replication is taking place, then first of all, both the strands will undergo replication. Right? Both the strands will undergo replication, and on both the parental strand, new daughter strand will be synthesized, new DNA will be synthesized in which the nucleotides are made up of nitrogen picking up from the medium which is N14. So this time the new daughter strand complementary to the parent strand will be synthesized having N14. So all the DNA of E. coli after first generation in light medium will convert from heavy to hybrid DNA. Hybrid DNA means what? 50% of the strand is heavy and 50% of the strand is light. The white color is denoting light strand and yellow color is denoting heavy strand. So in first generation of replication, you will see that entire DNA of E. coli has become hybrid. Correct? Now, how to check it that whether it has become hybrid or still it is heavy? So for that, you need to perform centrifugation in the medium cesium chloride like Hershey and Chase did. Now, what will happen? You know, in centrifugation on density basis, we separate the components, right? Now, heavy DNA automatically will be the DNA in which both the strands are having N15 will be automatically heavier in nature as compared to the DNA which is having one strand N15 and one strand N14. So hybrid strands will appear lesser in density as compared to the heavy DNA. So that we can analyze with the help of centrifugation done in cesium chloride medium. Now, next step will be to further see the method to further continue replication of E. coli for the next generation. Now this is what? Hybrid DNAs. Now each hybrid DNA is made to replicate for the next generation, means second generation. Again, if DNA is replicating semi-conservatively, then now both the strand will become parent, medium is still light. So first of all, both the parental strands will separate from each other. Now total there will be 4 parental strands, 2 heavy and 2 light. Correct? N15, N15, N14, N14. Now what will happen? New daughter strand will be synthesized on each parental strand, picking up the nitrogen from the medium, which is N14, following the complementary base pairing rule. So over here, N14 strand, N14, N14, N14. So automatically hybrid, initially after first generation of replication, 100% hybrid DNA were obtained. But second generation, you see beta, the hybrid DNA, the amount of hybrid DNA reduces to 50%. Heavy DNA is lost. Now there is no more heavy DNA, because now every time with every round of replication you are incorporating N14 strand. So automatically, slowly and steadily, with each round of replication, with each round of generation, what will happen? The amount of heavy strand of DNA will keep on reducing and the amount of light DNA strand will keep on increasing. So the number has reduced from 100 to 50% hybrid. See, this is hybrid and this is hybrid. Now these two, the ones which you see in the middle, this one and this one, C and D, which I have written. Oh God, it's not visible, I think. Yes, the C and D. Now C and D beta, they are light DNA, complete light. Why? Because both the strands are made up of N14. So 50% is now light DNA. Now again, if you make them replicate in for the next round, that means for third generation, again after 20 minutes, if you want to check that yes, whether if the DNA is replicating semi-conservatively or not. So now all the 4 strands will act as parent. Okay? Now what will happen? See, number of yellow strands have reduced. Only 2 yellow strands. 1, 2, 3, 4, 5, 6. 6 are white strand. 1, 2, 3, 4, 5, 6. So 6 white strands. 1, 2, 3, 4, 5, 6. Now still the medium is light, which is N14. So now following complementary base pairing rule, new daughter strands will be synthesized, picking up the nitrogen from the medium, which is N14. So every time light daughter strand will be produced on each parental strand. So now can you see that the amount of hybrid has reduced to 25%. And the amount of light DNA has increased by 75%. So initially 100% hybrid, 50% hybrid, 25. So every round, because you're not changing the medium, you're keeping it light. So you see in 3 generations only, the amount of heavy DNA has reduced to such an extent. Now if you keep on continuing the replication for more generation, automatically all the heavy DNA or the percentage of heavy DNA will become very, very less, and almost whole E. coli DNA nature will change from heavy to light. Understanding? So why this is so? Why this number is there? Because DNA is replicating semi-conservatively. One of the parental strand is old, one of the strand is new, complementary to the parental strand. This is known as semi-conservative replication, which was explained by Meselson and Stahl while working on E. coli. Now, you have just seen that what is semi-conservative replication. But now you need to check out the mechanism, that how this whole process of replication is taking place. So now next important topic is to study the mechanism of replication. For that, you need to first list all the enzymes. Now, but it is a energetically expensive phenomena, replication. Number of enzymes are involved for this process. This process takes place in case of prokaryotes in the cytoplasm, but in case of eukaryotes, this takes place inside the nucleus of, it takes place inside the nucleus of the cell. For eukaryote, I am repeating once again, for eukaryote, it is taking place inside the nucleus, but for prokaryotes, this process is taking place outside the in prokaryotes there is no nucleus, there is only cytoplasm. So in case of prokaryote, obviously replication has to take place in the cytoplasm. First of all, we are going to check out all the enzymes which are required for the purpose of replication, and then we will study the mechanism. Right? Chalo. So first very important enzyme is DNA polymerase. Beta, what is replication? Replication simply stands for copying of DNA from DNA. There is one DNA. From this DNA making more number of DNA. This is only replication, copying of DNA from the DNA strand. That means you have to polymerize DNA, correct? Replication simply means synthesis of new daughter strands of DNA, and new daughter strands means polymerization. So the most important enzyme for this process will be DNA polymerase, right? Now there are three types of DNA polymerase found in case of prokaryotes. Now we are going to study replication in prokaryote. So all the enzyme list which I am going to tell you is related to prokaryote. In prokaryote, the most important enzyme DNA polymerase is of three types. DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. Be it any type of DNA polymerase, their main function is synthesis of new DNA strand in 5' to 3' direction. That means all the three types of DNA polymerase have the property of 5' to 3' polymerase. So all three, they have 5' to 3'. That means new daughter strand is always going to be synthesized in 5' to 3' direction. Okay, beta, the speed of polymerization, the speed of the synthesis of DNA is very fast. In case of, if I talk about DNA polymerase 3, it synthesizes DNA strand per second, 2000 base pairs are added. 1 second में 2000 base pair का लंबा strand बना देता है. So there are high chance of error also. In 1 second, 2000 base pair, that means it is checking the complementary also, adding it and moving ahead also. Just in 1 second, 2000 base pairs can be added. So chances of error is also very, very high when the speed is such high. But one single error in replication can cause lot of disorders in the human body. So obviously any cell cannot take the risk of wrong DNA replication. Therefore, every polymerase along with polymerization activity, they also have proofreading activity to avoid error-prone replication in the direction 3' to 5'. It is known as 3' to 5' exonuclease. 3' to 5' exonuclease. This activity is specifically to avoid error-prone replication. Correct? So these two activities are common for all the three polymerase. DNA polymerase 1, also called as Korenberg's enzyme, apart from these two activities, they also have one special power called 5' to 3' exonuclease. That means DNA polymerase 1 can cut the nucleotide even from 5' direction. This is also very, very helpful. While I'll explain you the mechanism, I'll tell you that how this property is helpful in DNA replication. So the common two activities are 5' to 3' polymerase that helps in the synthesis of daughter strand. Now, to ensure that all the nucleotides which are added are complementary, no error replication is taking place. Each and every polymerase enzyme have 3' to 5' exonuclease. Now, specifically for DNA polymerase 1, you have to remember that they have one special power called 5' to 3' exonuclease. That means they can cut the nucleotide from 5' direction as well if required. Right? However, the most efficient and fast enzyme is DNA polymerase 3. So, it is required actually for DNA replication. If you want to ask that, ma'am, out of the three, which will be preferred by bacteria? So, answer will be DNA polymerase 3 because it is fast and more efficient as compared to the other two. Right? Apart from this, beta, next enzyme required is helicase, that helps in unwinding of the DNA duplex, because replication cannot take place in winded state. First of all, DNA has to unwind so that replication can start. Next enzyme is SSBP, single strand binding protein. This is required to keep the strand in unwinded state till the time replication is going on. It is required to keep the strand in unwinded state during the time of replication. Correct? Next enzyme required is ligase. Right now I'm just completing the list. While I'll explain you, I'll just tell about each and every enzyme and their use again. But right now I'm just writing the list, completing the list. So ligase enzyme is basically used to join the DNA fragment. Ligase activity is ATP based. Helicase also functions by utilizing energy. Ligase is also requiring energy for joining the DNA fragments. Next is topoisomerase, also called as DNA gyrase. This topoisomerase, beta, it is specially required to remove the supercoiling. I'll tell you how. To remove the supercoiling of DNA duplex. Okay? So these are the different enzymes which we are going to use during replication, and one more primase enzyme is also there, which is used to synthesize primer. It is used to synthesize primer. Now, how and when will let you know while I'll teach you the process, proper steps of replication. So now let's start the main mechanism of replication. The first step. Now see, this is your DNA. Okay. In case of, now we are studying about prokaryotic DNA. So prokaryotic DNA is single, double-stranded, circular. So this is how our DNA is right now. Okay. First step is to identify the region of replication, the origin point of replication. This is a DNA. Now, from where replication should start? There should be some origin point now. So the first step of replication is to identify the origin of replication. Origin of replication are specific sequences. They are specific nucleotide sequences which are identified by the enzyme helicase. Helicase goes and attack at ori to unwind the DNA duplex. Clear? Now, in case of prokaryote, on the whole DNA, there is only one ori. But in case of eukaryote, we have so many DNA, so there are thousands of ori. First step, to identify ori. Ori stands for origin of replication. So that the process can start from the right location. There should be no error-prone replication, and for that, the very first important thing is that replication should start from the right location. Suppose there is a race going on, right? So if you really want a proper winner, if you really want to check that who is the fastest person, then you will make all the participants stand on one line now. And then only you will start the race at the same time, and then you will see who has reached the final point first. Correct? Imagine if there is no start point, if there is no origin point, and some people are running like 2 kilometers behind, some people are then joining, then a random winner will come. Now you will not have a proper judgment that okay, out of these 10 people, who is the fastest. If you really want to have a proper judgment, then there should be some origin point from where all the participants are standing under same condition, then they are made to run at the same time. Then only you can judge then only you can take the right decision. Therefore, similarly, till the time we do not start the process from the correct location, how can we accept a correct solution, right? So, origin of replication, so that the process can begin from the right location. In bracket write 1 ori for prokaryote and multiple ori for eukaryote, right? Now, ori is recognized by helicase. So, next step, but before helicase activity, beta, there is one more thing. Okay, our E. coli DNA is circular. So, you just keep that in mind. But I will draw it like this for my convenience. Both the strands are anti-parallel to each other. Okay, right now, beta, what is the condition? The parental DNA which are about to start the process of replication. Repeating it once again. The parental DNA which are about to start the process of replication, initially their nucleotides are present in monophosphate or diphosphate state, which is a non-energetic state. So next step is converting the parental nucleotides into energetic form. That means to produce deoxyribonucleotide from deoxymononucleotide or dinucleotide. So next step is to produce deoxyribonucleotide triphosphate from mono or biphosphate. I mean to say, initially this is the DNA strand. Now both the strands will be complementary, right? Anti-parallel. Here will be deoxy, maybe ADP, CDP, TDP. Here the complementary TDP means initially the nucleotides, they are present in diphosphate state, but which is not energetically active. So replication process cannot start, right? So first step is that with the help of energy and in the presence of the enzyme phosphorylase, not in the form of ATP, in the presence of the enzyme phosphorylase and phosphoric acid. In the presence of phosphorylase enzyme, phosphoric acid is used to donate its phosphate to all the deoxyribonucleotide diphosphate so that they can be converted into their triphosphate states. So what will be the next step? See, here I am drawing little bit bigger. Right? So after phosphorylation, ADP will change to ATP, CDP will change to CTP, GDP will change to GTP. In front of GTP on the other strand will be CTP, CTP के सामने GTP, ATP के सामने TTP. That means you have made the strand now energetically expensive. There are two reasons behind it that why have we done this. Because now these nucleotides on the parental strand, first of all, they will act as the substrate for the new daughter strand synthesis. Plus, new daughter strand synthesis requires energy. So they will also provide energy because now they are present in energetic form. ATP is a source of energy, GTP is a source of energy. Here is ATP, here is GTP, here is CTP. All these are source of energy. So once we activate, this is called as activation of deoxyribonucleotide to produce deoxyribonucleotide triphosphate. They serve two purposes. The second step serves two purposes. What is the first purpose? The first purpose is acting as substrate for the synthesis of daughter strand, and second, they also provide energy for the synthesis of new daughter strand. Now, after the second step is completed, now comes the helicase enzyme. Helicase enzyme is now used to unwind the double helical strand, the helical strand of DNA. See, now this is the condition, right? Both the strands are anti-parallel, 5 prime, 3 prime, 3 prime, 5 prime. The nucleotides are now activated, ATP, CTP, GTP. So complementary, here will be CTP, GTP, TTP. Now, but right now they are in winded state. Replication process cannot start. So first of all, unwinding is required. So helicase enzyme will come and will pull both the strands like this. Okay, imagine there is a ponytail. The ponytail now, which in, I don't know how, in braided form. The ponytail is in braided form, right? Now, if suppose I am having a braided ponytail and if I want to open it, so one way is that I properly unwind it. One way is that I held the two ends and I pull it. So helicase will unwind the DNA by just pulling it but not properly unwinding it. As a result, the lower strand will become unwind. But due to applying pressure, unwinding above the lower region will become more winding above the region will become more complex. Suppose this itna portion got unwind, but due to helicase activity above this, it will become super coiled, highly coiled, right? So now when helicase will come, it will require energy, it will unwind the DNA, but it is going to pull the strands from below. So lower portion will get unwind, but above portion will get super winded up like this. And lower portion will open to form a Y-shaped replicating fork. So this is 5 prime, 3 prime, 3 prime, 5 prime. Due to helicase activity, Y-shaped replicating fork is produced, exposing the nucleotides present in triphosphate state. Now these nucleotides are ready to act as substrate, as well as they are ready to provide energy for the synthesis of new nucleotidic strand. Now synthesis is always going to occur in 5 prime to 3 prime direction. Which enzyme is going to be useful? DNA Pol 3, because it is more efficient, it produces less error. Okay, so on one of the parental strand, this parental strand, replication will occur in this direction. So that 5 prime will be over here, anti-parallel nature has to be proved, and on the other strand, replication will take from above to below. So that 5 prime should come over here. Do you know this thing? Have you understood the reason? The reason is that even further daughter strands should also be anti-parallel to each other. In front of 3, there should be 5. In front of 5, there should be 3. So here is 3, so here should be 5. Here is 5, here should come 3. Understood? Okay. Now, after helicase activity, next comes the primase activity. Next step is addition of primer. Now, what is primer, beta? Primer is a short oligo. Oligo means 2 to 9 nucleotides free in the cytoplasm of prokaryotic cell. They randomly join with the help of the enzyme primase to form a short polymer of nucleotide. So, primer is a short oligonucleotide strand produced with the help of enzyme primase. Okay, now suppose in the cytoplasm, obviously there are so many free nucleotides. All these are nucleotides, random, DATP, DCTP, DGTP, random nucleotides. So now these random nucleotides, in the presence of the enzyme, they will be made to join in a strand-like manner. So all the nucleotides have attached themselves in random manner. There is no picking up. Near about 2 to 9 long strand is being produced with the help of primase. This strand is known as primer. Now this primer goes and get attached over here. See, this was the condition now. This was my strand. Y-shaped replicating fork is produced. Now one strand is 3 like this. Okay. Now on this parental strand, which I am marking as A, this is A parental strand, this is B parental strand. Now on A parental strand, beta, the primer is going to get attached over here. And on B parental strand, primer is going to get attached over here, which marks the beginning of replication. That means at the 5 prime end of the daughter strand, primer is going to get attached. So primer, beta, attaches itself at the 5 prime end of each daughter strand. Now why this primer, this random primer is required? Even the primer is sometimes not even following the complementary base pairing. So why this wrong primer is attached? only because DNA polymerase 3 cannot initiate the replication. DNA polymerase 3 need a small ladder. DNA polymerase 3 enzyme, which is required for the synthesis of new daughter strand, cannot initiate the process. Hence, a primer is attached, a small primer comes and gets attached. So, now DNA polymerase 3 will come, sit on the tip of the primer, start reading the nucleotides and start joining the new strand in complementary manner from 5' to 3' direction. So primer attaches because beta DNA polymerase 3 fails to begin the process. Hence, a primer is required to attach in the beginning so that now DNA polymerase 3 can join the process and let the whole replication follow till the top. Okay, one more thing I just forgot to tell you, the role about SSBP. See beta, once the unwinding is done, now to keep the strands in unwinded state over here, SSBP protein is required. So role of SSBP is to keep the DNA strand in unwinded state till the time replication is done. Now over here, see, replication is done. Now what will happen? SSBP will be removed. As soon SSBP gets removed, this strand, it gets winded up. Here it gets winded up. Now what will happen? What is the next step? Check it out. So what is the condition now? See, SSBP got removed. Above there is supercoiling. If SSBP gets removed, now this much DNA got replicated. Here there was a short primer. Here there was a short primer. On top of that, new daughter strand was synthesized which just got winded up, so that, why? Because SSBP got removed. Now further replication of this portion need to be done. So first, supercoiling need to be removed, which cannot be done by normal helicase. So now topoisomerase, gyrase enzyme, topoisomerase called as gyrase enzyme will come to remove supercoiling. Now once the supercoiling will be removed, so what will happen? Now again SSBP will come. Now this is the lower condition. Wait, let me draw it over here so that my hands reach. Somewhere in the beginning there is a primer attached. Here is the primer. And over here is the primer. Right? Okay. Now what will happen? Now above DNA has unwinded. Here SSBP is attached. Now supercoiling has now moved further upward. This is the now current scenario. Till here replication is done. Now further replication need to be done. This is 3', 5' of the new. This is 5'. Right? Now beta, what will happen? One of the strand where primer was attached in the lower side, this is the primer, where the primer was attached on the lower side, that DNA will continuously replicate. But the other strand where primer was attached in the beginning, here, when the primer was attached in the beginning, now here you can see there 3' is over here. Now below replication is not required now. Replication need to again start from the back. That means again we need to begin. That means again a first primer is required so that further replication can take place. That means one of the strand is continuously synthesized, but another strand is discontinuously synthesized, as on that strand again and again primer need to be attached. So during replication, beta, one of the strand is continuously synthesized. Daughter strand is continuously synthesized, which is called as leading strand. Whereas the other strand, due to continuous addition of primer, is synthesized in fragments. These fragments are called as Okazaki because it was discovered by Okazaki, and such strand is known as lagging strand. Clear? Yes or no? Now, okay, later on the lagging strands, the fragments of the strand are later on joined with the help of the DNA ligase to again complete the second strand also. So first write this point and then I will tell you more thing. Later on, beta, these fragments are joined with the help of ligase enzyme. Again, it is ATP requiring enzyme. Now, okay, complete process is done. But you remember primers are still attached. So suppose this is the condition. Right? For example, over here, whole DNA replication, now everything is done. Like ligase has functioned. Now in the beginning, here one primer is attached. Here so many primers are attached due to discontinuous synthesis. Now these primers need to be removed from 5' direction. Now these white colored are beta primer. Here only one primer is there, and on the other strand there are many primers. Now, till the time you do not remove the primer, DNA replication is not complete, and primers are attached at 5' end. So, these are wrong nucleotides which has to be removed, and the wrong nucleotides have to be removed from 5' end. That means we require 5' to 3' exonuclease, which is present only with DNA polymerase 1. So now DNA polymerase 1 will come. Now DNA polymerase 1 is required, beta, to remove primers. Yes or no? They are required to remove the primers from 5' to 3' end with the help of its. Now which activity it is going to use? With the help of its 5' to 3' exonuclease activity. Right? So now 5' to 3' exonuclease activity is going to be utilized by DNA polymerase 1 to remove the primer. Gap will be created. Now those gaps will be filled by DNA polymerase 1 itself using its 5' to 3' polymerase activity. New strand complementary to the parent strand will be synthesized, and thus DNA replication will be completed. So this is how the whole mechanism of DNA replication takes place. A quick revision: the first step is to look for the origin of replication so that helicase can go and unwind the DNA. Second step is to activate the nucleotides of the parent DNA so that they can serve two purposes. First, they can act as a substrate, and second, they can provide energy for polymerization. Third, now of course, helicase will come, unwind the DNA. To keep the DNA in their unwinded state, helicase SSBP will come and will get attached. Now synthesis will take place in 5' to 3' direction, but before that primer need to be attached as DNA polymerase 3 fails to start the process. Correct? Okay, now primers are attached, polymerization is done. One strand is continuously synthesized, another strand is discontinuously synthesized called as lagging strand, and those fragments are called as Okazaki, which are later joined with the help of the enzyme DNA ligase. At last, all the primers need to be removed with the help of 5' to 3' exonuclease activity of DNA Pol1. The gaps thus created are again filled by the same enzyme utilizing its activity 5' to 3' polymerase this time, completing the whole process. So DNA is replicating semi-conservatively with the help of so many enzymes. I hope you all have understood the whole mechanism, whole process of DNA replication. So for today, we are going to keep it till here. Please read it from NCERT once as well as go through the notes plus complete the questions which are given to you. So with this, I would like to say thank you. Have a nice day. Bye-bye everyone. Take care.